Our next membrane-bound organelle is what's called the Golgi apparatus. And the Golgi apparatus is a very, very important organelle that's found in all cells. And it's important because it packages, processes, and sorts proteins. I like to think of the mnemonic PPS to help me remember this. So it's package, process, and sort. So it's a group of stacked and flat membranous sacs. It modifies, concentrates, and packages proteins and lipids that it receives from the rough endoplasmic reticulum. So in the rough endoplasmic reticulum, that is where the protein is synthesized. So it's responsible for protein synthesis. Once that protein is made, it is then packaged. We can say modify, concentrate, package. All of those kind of mean the same thing. And it processes it. So it gets it ready to then go to its final destination. So in this case, what we see here is a transport vesicle from the rough ER. So that transport vesicle contains a protein. Once it enters the Golgi apparatus, it's then going to do various things like maybe add a lipid to it, maybe add a carbohydrate to it. Let's say that it's going to be a protein receptor to be embedded as an integral protein in the phospholipid bilayer. It's then shipped via a transport vesicle that travels to the membrane where it's embedded. So there's lots of different things that can happen to that protein in the Golgi apparatus. So it transports the vesicle. The proteins or lipids are taken inside, further modified, tagged, sorted, and packaged. So it's kind of like a traffic director. So I like to think of the Golgi apparatus as either the post office or the FedEx station. In both cases, those two places, like the Golgi apparatus, are going to take your package. They're going to modify it. But in the Golgi apparatus case, that modification doesn't mean put a stamp on it. It means maybe to add a glycogen strand to it or to further fold it as a protein into a quaternary structure. Once it does that, it then sends it to its final destination. So instead of shipping it like the FedEx station would do, it sends it via a transport vesicle to its final destination. So the combination of the endoplasmic reticulum, the Golgi apparatus, is what we like to refer to as the endomembranous system. So the vesicles containing the proteins bud off from the endoplasmic reticulum. Then the proteins are modified in the Golgi apparatus. There could be a sugar group added to them. Maybe there's a phosphate group that's added. Then they're finally sorted. They're distributed or addressed to their final target. So here shows some possible destinations from the Golgi apparatus. Maybe they're shipped out of the cell, and that would be exocytosis. Maybe they become part of the membrane, and that's what happens here. Or maybe they're set for destruction, so they go to a lysosome. So there could be proteins for secretion via secretory vesicles. They could be destined for membranes, or they could be digested, or they could be committed to digestive enzymes, which is what lysosomes contain, and what we're going to talk about on our next slide. So this slide is showing us a magnification of a lysosome. Kind of think Lysol when you see lysosome. It's a membranous bag containing digestive enzymes, usually enzymes called hydrolases, And they're considered safe sites because they isolate harmful intracellular material and digest them from the rest of the cell. So they could digest ingested bacteria, viruses, or toxins. Another thing they could digest is non-functional organelles. They could be responsible for metabolic functions like releasing glycogen. Remember that glycogen was a polysaccharide that you learned about in chapter 2, that glycogen is broken down into glucose. So let's say that glycogen, as you recall, is stored in skeletal muscle as well as the liver. The hormone called glucagon, released from the pancreas, is going to be released when your blood sugar is low. It's going to tell the glycogen to be broken down into glucose. And that glycogen is broken down into glucose in a lysosome. Another function is the release of calcium from the bone, which happens when our blood calcium is low. We'll learn more about that in chapter 6. And finally, the last function is that it could digest itself, which is called autolysis. So this slide is showing us these membranous organelles. So we talked about the endoplasmic reticulum, the Golgi apparatus, secretory vesicles, and lysosomes. So together, these membranes and organelles are all going to work together to produce, degrade, store, and possibly export different molecules, or they could degrade potentially harmful structures. So the smooth endoplasmic reticulum is important for lipid metabolism. The nuclear envelope is what is going to surround the nucleus, and it only allows for the export of what's called messenger RNA, which we'll be talking about later on in the semester. The ref endoplasmic reticulum's function is going to be that of protein synthesis. The Golgi apparatus is going to package, process, and sort proteins. So it receives the proteins from the rough endoplasmic reticulum. The transport vesicle it can transport a whole lot of molecules in different directions. The lysozyme has hydrolases in it that can be for metabolic reactions. It can be for autolysis. We see another secretory vesicle, in this case, is going to secrete molecules from that cell. So this would be responsible for exocytosis, remember, as a type of active transport. And we've already talked about the plasma membrane. And it's very important to remember that the plasma membrane has the characteristic of selective permeability. It only allows certain things in and out if it has proteins for that, which form channels.